Hello, everyone. My name is Alex Hoischen, and I'm an associate professor at the Human Genetics Department in Nijmegen, the Netherlands. And it's a great pleasure to talk to you about the identification of hidden structural variants with optical genome mapping. What I would like to share is an experience in using optical genome mapping by BioNano for roughly 20 months by now. Despite the lockdown measures in our own laboratory, we still managed to analyze a total of 250 samples over the last 20 months or so. The huge focus has gone into using optical mapping to compare it to our routine workup for cytogenetics. So that is usually a mix of karyotyping fish and CNV microarrays. And we did that for two applications: leukemia samples and samples with constitutional cytogenetic aberrations. What we have done since then are new studies. For example, we looked at several samples with known repeat expansions. We fine-mapped cases with balanced translocations. And we also have several lines of research where we're interested in very low-level mosaicism for structure variants. And again, optical mapping is a very suitable tool for that. But what the main focus of today's talk will be some of our unsolved rare disease research cases. And I'll guide you through some of that work in the remainder of the talk. Just getting back to the very first 2 points, our clinical utility studies and the proof of concept studies we've done so far, two of my great collaborators are presenting that work in this cytogenomics webinar series. That is Dr. Leila El-Khattabi talking about constitutional aberrations in 85 samples, and my former postdoc and now independent scientist in Oulu, Finland, Dr. Tuomo Mantere, he will present our work on 52 leukemia genomes. So I hope you'll attend those seminars as well, and if you're interested in more underlying data, I would refer you to the two bioRxiv preprints that we already have posted. But now to the main topic of today's seminar. BioNano may really enable us to find novel research findings. And those in particular should be structure variants that so far remained refractory to any detection by previous technologies. Some people also refer to those as being in the camouflaged parts of the human genome. And what kind of rare disease cases are suited for this? Think, for example, about families or cases where you already know the disease gene locus for sometimes quite a long time, but so far sequencing has not succeeded in identifying the disease-causing mutation. Other cases we've included so far are classical recessive disease cases where a mutation was found by sequencing on one of the two alleles, but so far the second mutation remains undiscovered. And there again, speculation may be that the second mutation is a difficult to identify structure variant. What we also focus on are hidden structure variants that occur de novo. We know that the de novo mutation rate for structure variants is seemingly quite low. So if you go for patient-parent trios, we should be able to also identify those systematically using optical mapping. Where do we retrieve our samples and case series from? That is usually directly from several collaborating groups in our very own department, so the medical center of the Radboud University, where next to my own group, we have 17 other PI groups that work, at least a lot of them work on rare diseases. Then we are engaged in several European and national consortia that also focus on rare disease research, but we're also open to other collaborative studies, and I'll show you more details about that. Just the high-level summary of all the research projects run so far. Altogether, we have run 20 research projects, and we have extremely interesting leads for at least 5 out of those 20 projects. Those can be projects that consist of just one sample in some occasions, but sometimes a series of up to 10 samples also for other projects. And I thought the best would be to highlight 2 cases where we recently discovered interesting findings. Here's the first case. And this is really illustrative for a larger series of experiments that we usually do. The model disease that we study a lot is severe intellectual disability. And that, especially in the Western and outbred population, is very often a sporadic disease and very often is caused by de novo mutations of all kinds. De novo point mutations, as well as de novo structural variants. And to find those more systematically, we study patient-parent trios a lot. So affected patients and the unaffected parents are run in parallel. And in a small series of samples that I present today, we have done all the previous assays already. So these are patient-parent trios that underwent CNV microarray analysis, exome sequencing, and genome sequencing, and most recently, even long-read genome sequencing. And we used 15x PacBio coverage for that. And unfortunately, none of these previous assays identified a disease-causing or likely disease-causing mutation. And therefore, the very simple and straightforward question is, can optical mapping identify the so far hidden de novo structural variant, and may that variant even be linked to disease and may therefore be a novel candidate variant? So what we've done is the series that I'm describing here are just 5 trios. And actually, we only identified and validated a single de novo structure variant in 1 out of the 5 trios. And I'll just guide you through the filtering process that we underwent. So this is a typical trio, and I show you the structure variants called in the proband, the patient. So we usually find 6,000 to 7,000 structure variants per case. By just very simple filter strategies, for example, by only looking into the private structure variants, hence all the structure variants that don't overlap with structure variants in the 200 controlled genomes available to us, we already reduce the number of structure variants dramatically, usually to something in the range of 30 to 50 events per case. But then, if we use parental samples in addition, we can just click once more and look for all the variants that are not present in mom and dad. And in this case, that was only exactly 1 aberration, 1 deletion that is seemingly de novo. Before I show you details of this 1 deletion, I just wanted to make 2 important points. What do we already learn from this exercise on its own right? This already teaches us that de novo mutations for structure variants that are larger than 500 base pairs in size, that de novo mutation rate is probably relatively low, similarly low to what we expect from array and genome sequencing. It furthermore also shows us that optical mapping has an extremely low false positive rate, because de novo mutations would enrich for false positive findings, or the filter approach of de novo mutations. But by very simple filters, we really just end up with 1 true event and not a single false positive. So that is very encouraging in terms of quality of the optical mapping. So this is the 1 de novo event that we identified, and it ended to be in one of the most complicated regions of the human genome. This is a region in chromosome 17, Q21. And that is an extreme, here in this region, the human genome shows a large cluster of segmental duplications. And what we identified is that this individual, the patient, carried a paternally inherited duplication of this locus, but in addition, a de novo deletion just exactly downstream of that, most likely affecting and disrupting a gene known as NSF. This deletion was validated simply by designing breakpoint spanning PCR primers. And here you hopefully appreciate that there's a successful band of the mutated allele present in the patient, but not present in mother nor father. This PCR fragment was then sequenced using PacBio amplicon sequencing, and this validated that this truly was a de novo deletion of 121 kb in size. So a pretty large deletion, And again, it was not identified by any previous technology. What are now the details of this deletion? As I said already, this happens in the 17q21.3.1 region of the genome, really just affecting the segmental duplications that are downstream of the colon diffuse microdeletion syndrome region. Most likely, this disrupts the gene NSF. However, interpretation of that is not trivial because NSF just upstream has a pseudogene NSFP. What is known about these two genes that it's extremely copy number variable in the general population. So there have been studies that propose that there's between 2 and 8 copies of NSF, or likely the combination of NSF and NSFP. So the big question is, while we now identify the very first de novo structure variant in this case, it is intriguing to speculate whether the haploinsufficiency of NSF, where this event most likely leads to, could potentially be a cause of disease. And that really requires further research. So if anyone is aware of deletions of NSF, I would be very curious to get in contact. There is, however, very intriguing recent work, because point mutations in NSF in a study from Japan have been linked to an epilepsy phenotype. So different phenotype, but also the mutations are different. They are point mutations, and the authors of that manuscript explain that they most likely affect the gene function and the protein function by a dominant negative effect. So still the question would be whether simple haploinsufficiency of NSF could cause a disease, but we may have the first case here, so hopefully we identify additional cases in the near future. I brought a second example today to illustrate what we can identify by in terms of structure variants by optical mapping. And that is a collaboration with an old colleague of mine, Roland Kuiper, who now works in Utrecht, also in the Netherlands, in the Prinses Maxima Center for Pediatric Oncology. And one of Roland's interests is a very rare but aggressive form of cancer that is usually diagnosed in childhood, and that is known as atypical teratoid rhabdoid tumors. Usually those present as brain tumors, but can also affect other parts of the central nervous system. The cause for these tumors to occur lay in a gene known as SMARCB1. In quite a lot of cases, there's somatic mutations in SMARCB1 happening early in development, but in roughly 1/3 of all cases, there's already a predisposing germline mutation in this gene, and a second somatic hit then happens in the cancer itself. So Roland and his clinical colleagues have studied a Dutch family with a strong suspicion of a germline mutation in SMARCB1 already, based on the phenotype identified in one of the children. However, so far no mutation was identified, despite huge efforts. And the previous efforts included diagnostic Sanger sequencing, MLPA, and whole exome sequencing, and none of that identified a mutation in SMARCB1. In addition, Roland's team also applied whole genome sequencing on research basis, but again, failed to identify the disease-causing mutation. So therefore, Roland sent an EBV cell line to us. We isolated ultra high molecular weight DNA from it and performed optical mapping. Similar to the filtering strategies that I've shown you before, we just look at all the rare variants that we identify in this genome. And here you see a list of events. So it's 12 insertions and 20 deletions. In total, 35 rare structure variants in this genome. And then we obviously already had an extremely good candidate locus. What you see here is an event that was called exactly in the gene SMARCB1. So that's one of the 35 private structural variants in this person's genome. If I zoom into the details, you will see here that there is an insertion of close to 2.8 kilobase pairs that also creates one new label that is not present in the reference, but it is present in the assembly of the patient's molecules that we studied. So this is an insertion that happens between, most likely between the introns 1 and 3 of SMARCB1. And we dared already to speculate on this. Highly likely this is a so-for missing germline mutation that gives a very strong predisposition to ATRT. We did not know the exact sequence of this inserted material. So therefore, Roland's team tried to validate by PCR various forms of PCR and resequencing, but unfortunately that remained unsuccessful. So leading to the speculation that it must be a complex part of the genome inserted in this very spot. So therefore, at the end, we decided to perform PacBio HiFi sequencing. So we created a library of 20 kilobase pairs and sequenced the genome with an average of 8-fold coverage. And that validated the insertion, but also made it more refined. So thanks to the sequence context we now get from the long-read sequencing, we now know that this is an exactly 2,763 base pair insertion in intron 1 of the SMARCB1 gene. If you look at the sequence context, we now also understand that it is a retrotransposon insertion with quite a repeat structure at either end of the inserted material, and that is the likely explanation why PCR and even genome sequencing by Illumina failed so far to identify this event. I think this is a really tough region to sequence, and the context of it does not make it easy to fish it out and see it. So as such, we now believe that this truly is the validated germline structure variant that gives the strong predisposition to ATRT in this family. However, how exactly an intronic retrotransposon insertion in an intron leads to a loss of function allele is still further studied by work on RNA level by Roland Kuiper's group. But interestingly, you could now ask yourself whether there would be more cases where we have these kind of jumping retrotransposons that may disrupt genes. And seeing the difficulty of identifying this one by other classical means, you may ask yourself, how often does that happen? And how often does it happen in so far unsolved cases, even after exome and genome sequencing? So after highlighting these two cases, I just want to finish with a little summary. From all the data we've generated over the last month, I see a tremendous potential for optical genome mapping to replace classical cytogenetics, and that is in particular karyotyping, CNV microarrays, and FISH. For the research cases I've presented today, I hope I gave you a first flavor that the technology can identify hidden structure variants, and in particular variants that have been missed by so far available NGS approaches. I just tried to highlight that with the two examples, a de novo deletion that affects the gene NSF1 and a retrotransposon insertion in the gene SMARCB1 as a likely pathogenic germline mutation. Very extremely recent research findings include several other findings. So we just recently identified a hidden inverted duplication in a family suffering from genetic forms of blindness, in which the locus was known for more than 20 years, but the disease-causing mutations on DNA level remained unknown. We've also started to see the first de novo structure variants in clinically defined syndromes, and we're following up on those. We also managed to fine map balanced translocations to understand the breakpoints in greater detail and understand whether balanced translocations either disrupt genes or disrupt the regulatory regions of important downstream genes. And for some samples, we now have been able to measure long repeat expansions and repeat expansion disorders that so far by classical means were unmeasurable. To sum it up in simple words, I think optical genome mapping by Bionano offers clinical and research benefits and is already enabling us to really live the dream of a cytogenetics revolution. So that is extremely exciting to us and hopefully to many others. Finally, I'd like to acknowledge everyone involved in the studies I presented today. That is, of course, Roland Kuiper's group from Utrecht, everyone at Bionano, but in particular also Tuomo Mantere and Cornelia Neveling from our team in Nijmegen. And I'd like to thank you for your attention and be happy to take questions. Thank you very much.